Good morning, students. Today's topic is fructose metabolism. All of us knows that fructose is a ketose sugar found in fruits, honey and sucrose. Soft drink and corn sugar also contains high fructose content, and fructose is sweeter than sucrose. Fructose is rapidly absorbed from GIT by facilitated diffusion. So this is the actual metabolism of fructose. Fructose from the diet is first converted into fructose 1-phosphate with the help of enzyme fructokinase in the liver. So this fructose 1-phosphate is later on broken down into two 3-carbon compounds, that is glyceraldehyde and dihydroxyacetone phosphate. Now this glyceraldehyde later on phosphorylated with the help of enzyme triose kinase to form glyceraldehyde 3-phosphate. Now both these compound glyceraldehyde 3-phosphate and dihydroxyacetone phosphate then enters into the glycolysis. So finally fructose enter into the glycolysis in this way. Fructokinase is an enzyme found in the liver and it has high affinity for fructose than hexokinase. Fructokinase phosphorylate the fructose at first position to form fructose 1-phosphate, whereas hexokinase phosphorylate the fructose at 6th position to form fructose 6-phosphate. So this is the difference between fructokinase and hexokinase. One more important thing about fructokinase is that fructokinase is not dependent on insulin. Therefore, it is more rapidly utilized as compared to glucose in normal persons as well as in diabetic persons. So theoretically you can say fructose is better utilized in patient with diabetes mellitus because fructokinase is not dependent on insulin. But in experiment it was found that fructose was more deleterious in diabetic patients. It causes atherogenic effect, it causes energy deficiency inside the cells, it causes hyperuricemia and it also causes hypoglycemia. So we will see how this fructose causes these effects. First, about atherogenicity. Fructose is more rapidly utilized as compared to glucose. Therefore, it stimulates the fatty acid synthesis. It increases the sugar level. It enters the glycolysis pathway, and through the glycolysis pathway, it forms pyruvate acetyl CoA and then stimulates the fatty acid synthesis. Fructose also raises the serum triglycerides level. Fructose increases LDL cholesterol level in the blood. So all these effects of fructose are due to its rapid utilization and they are atherogenic and harmful for an individual. Second effect of fructose is causes energy deficiency inside the cell. When fructose is rapidly utilized by the enzyme fructokinase, it converts the fructose into fructose 1-phosphate. Therefore, large amount of phosphate levels inside the cell are utilized for conversion of phosphate to fructose 1-phosphate. Therefore, there is deficiency of inorganic phosphate inside the cell and this causes decreased conversion of ADP to ATP because conversion of ADP to ATP requires inorganic phosphate. So in this way, fructose leads to energy deficiency inside the cell. Rapid utilization of fructose also increases the uric acid level of the blood. This uric acid, all of us know that, is formed from breakdown of AMP. When there is deficiency of ATP, This ATP act as a inhibitor of enzyme adenosine deaminase, and this adenosine deaminase is the main enzyme required for the catabolism of AMP. In the absence of inhibitor ATP, this adenosine deaminase is stimulated and it breaks more and more AMP into uric acid, leading to hyperuricemia. So this is the summary. Rapid utilization of fructose leads to more rapid formation of fructose 1-phosphate from fructose. Therefore, inorganic phosphate level inside the cell decreases. Hence, conversion of ADP to ATP get slowed down. Deficiency of ATP removes the inhibitory effect on enzyme adenosine deaminase, which is the main enzyme in the breakdown of AMP. Therefore, more and more AMP is converted into inosine monophosphate, which later on get converted into uric acid, and hyperuricemia is the result. When fructose 1-phosphate is not utilized, it get accumulated in the liver and it inhibits a process called as glycogenolysis. When the glycogenolysis is inhibited, the patient often goes in hypoglycemia because his blood glucose level falls in the post-absorptive state. In the post-absorptive state, the main supplier of glucose is breakdown of glycogen, that is glycogenolysis. Now, in the muscles, fructokinase is absent. Therefore, in the muscles, fructose is utilized with the help of enzyme hexokinase, which convert the fructose into fructose 6-phosphate. Though fructose is mainly metabolized in the liver, fructose is also found in large quantity in seminal fluid. Fructose is utilized as a source of energy by spermatozoa. Here, the fructose is secreted by seminal vesicles. Clinical significance of fructose in seminal fluid. In some persons of azoospermia means decreased sperm count, there may be block in the duct. In such person, if we estimate the fructose and if we found fructose in the seminal fluid, then we can say that the block is above the level of seminal vesicles. If the fructose is absent, then we can diagnose that the block is below the level of seminal vesicles because fructose comes from seminal vesicles in the seminal fluid. High fructose level of blood also causes aging and myocardial infarction. Fructose causes glycation of proteins. That means glycation of proteins means addition of sugar to the proteins. So glycation of proteins is 10 times faster by fructose than glucose. Hence, there is rapid damage to the proteins. Also, fructose causes decreased antioxidant effects. Antioxidants are the compounds which prevent our body from oxidative stress. But this fructose decreases the level of antioxidants in the body. Therefore, oxidative stress in the body increases and this will lead to ischemic heart diseases. Now come to inborn error of fructose metabolism. Just like galactose metabolism, it has two inborn error of metabolism. First is hereditary fructose intolerance and second is fructosuria. We will see hereditary fructose intolerance first. It is an autosomal recessive disease seen in 1 in 20,000 birth. The enzyme defect is aldolase B. So due to deficiency of this enzyme aldolase B, fructose 1-phosphate is not broken down into glyceraldehyde and dihydroxyacetone phosphate. Therefore, the fructose 1-phosphate get accumulated in the liver, and this deficiency is found around 6 months of age when sucrose is given for an infant in the diet which contains fructose. So when fructose 1-phosphate is accumulated in the liver, it inhibits the enzyme glycogen phosphorylase. Glycogen phosphorylase is the key enzyme of glycogenolysis. So glycogenolysis is inhibited. When glycogenolysis is inhibited, glycogen is not converted into glucose and infant have repeated attacks of hypoglycemia. He may also have vomiting, loss of appetite, fail to thrive, means his growth hampers, and accumulation of fructose 1-phosphate in the liver leads to hepatomegaly, that means increase in the size of liver. At the same time, liver cells are damaged and the patient will have jaundice. If this damage is progressive, then death will occur. How will you diagnose? Diagnosis was done by estimating the fructose in urine by Benedict test, and if we withdraw the fructose from the diet, the symptoms are immediately relieved. The next inborn error of metabolism is fructosuria. It is a benign, that means less harmful condition. Here the enzyme deficiency is fructokinase. So enzyme fructokinase is deficient in liver. Therefore, free fructose is not converted into fructose 1-phosphate. The level of fructose in the blood increases. This fructose is filtered into the kidney and excreted into the urine. It also gives Benedict test and Selvanov's test positive. This fructosuria is seen in 1,30,000 live birth. So this is about the fructose and its inborn error of metabolism. Thank you very much.